Hello everyone, my name is Dr. Haytham Tayyib. I'm an Associate Professor of Neurology, Neuropsychiatry and Behavioral Neurology and Clinical Neurophysiology at the King Abdulaziz University in Jeddah. It is a pleasure to be able to speak to you today at the beginning of this scientific session put together by the Saudi chapter of Cognitive and Behavioral Neurology. This new chapter is led by a group of excited and enthusiastic experts in the field who are ready to move the field and the community to the next level by working with all of you, inshallah. Today, I will be speaking about localization in cognitive neurology. This is a title that arose from popular demand. If you have participated in the needs assessment survey sent out by the chapter, this is one of the areas that you asked us to talk about. The main purpose of this talk is to remind us that cognitive and behavioral neurology is a discipline with strong grounding in neuroanatomy, neurophysiology, and the neurosciences. It is definitely more than a bunch of scores on scales. It's more than the MoCA and the TORCA and the Fulstein. These tests have their specific uses that are important and they have cutoff scores and everything, but those do not replace the basic approach that is the soul and spirit of cognitive neurology. The way I like to put it is that cognitive and behavioral neurology is neurological, meaning that it must follow the logic of neurology. It must begin with analyzing clinical phenomena and answering our time-cherished questions of where the lesion is before considering what the lesion is. Losing sight of this logic is a deviation from standard clinical practice and risks losing sight of what the important problems may be. All right, so in demonstration, let's start discussing memory functions. And yes, it is memory functions as a plural. People may think of memory simply as the 3 words or 5 words that you give your patient so that they can blurt it out to you in a few minutes, but that is a very narrow understanding of memory. All right, so here are the 2 divisions of memory. There is short-term memory and long-term memory. When we talk about short-term memory in the clinical world, we're basically referring to working memory, the memory that you keep in your mind for a little bit as long as you want to use it, and then you just purge off of it and you no longer remember it. And this includes things like phone numbers or certain images that you temporarily keep in what we call the buffer. And this is basically localized in the lateral prefrontal cortices, with verbal memory being on the dominant hemisphere and nonverbal memory being in the non-dominant hemisphere. And that is basically all we call short-term memory. And that's it, that's all. Everything else is long-term memory. What happened yesterday is long-term memory, and what happened 20 years ago is long-term memory. You can call recent and remote long-term memory, but both are long-term memory. And of course, recent long-term memory is more liable or vulnerable to being affected in disorders, genuine disorders of memory. That is what we call Rebo's law. There are 2 major divisions of long-term memory. There's declarative or explicit memory and non-declarative or implicit memory. Implicit memories are procedural memories, memories that you cannot really transfer to someone else as knowledge. And we won't be talking about them, but these are mainly motor memories and emotional memories of subcortical localization. We will discuss a little bit more explicit memories, memories that you can say and transfer as knowledge to someone else. And these are also of 2 types: semantic memory, which is encyclopedic memory that you can hold about objects and places and people. And these are non-time bound and it's just knowledge. Whereas episodic memory is time bound with content and context. So someone did something at a particular time, and so that is an episode. And both have 2 different localizations in the brain, and both are affected by different clinical syndromes. So Alzheimer's disease, for example, is a disorder of episodic memory, whereas semantic dementia is a disorder of semantic memory. Of course, semantic memory gets affected in Alzheimer's disease later on, but we're talking about the earlier stages here. So where is episodic memory localized? We know a lot about episodic memory from patient HM, that patient who had bilateral hippocampectomies because of refractory epilepsy. He had a surgery, and that caused him to have complete anterograde amnesia. He could no longer store information, and he had some retrograde amnesia as well. So that case demonstrated the importance of the hippocampus and of its associated circuit of PAPES. The way we encode episodic memory is dependent on this circuit. Information enters into the hippocampus through the parahippocampal gyrus and then is reiterated through the circuit over and over to encode it, to store it. So what happens at the micro level when that recurrent activation takes place is that you have a series of synapses that get strengthened when that activation, when the information is reiterated in the circuit, and so this causes encoding. What happens exactly is that glutamatergic receptors basically multiply. You first stimulate NMDA receptors, and that leads to a cytological intracellular cascade that leads to more AMPA receptors being expressed at the surface, and therefore the postsynaptic membrane being more excitable, a process well known as long-term potentiation, and that is how you remember things. And if that's how we encode memory, how do we retrieve them, and where are they stored? And here are the answers. We retrieve them basically using our frontal lobes. The frontal lobe is the clerk that goes and opens the storage areas of memory to retrieve the memory that you want. And here we're making a distinction between the mesial temporal lobes as basically the keys to our memory stores, and the frontal lobes as the retrievers of the memories from the memory stores. The frontal lobes are also important in registering memory. So the stage when you're giving the patient your 5 words, the frontal lobes work in the beginning to register the words, and the frontal lobes work in the end to retrieve the words, and in the middle, the mesial temporal lobes work to store and encode the words. Where are they stored? They are stored in the sensory cortices, basically. If you look here in this column, this is sensation. The occipital lobes are activated when you actually see something, and the temporal lobes are activated when you hear something. And this column represents the process of retrieving memories. So this is not perception. Now you are remembering something you've seen before, you activate visual areas. You're remembering something that you've heard before, you're activating temporal areas. And this is all the keys to this network of activation that are memory specific are bound in the hippocampus, and that's basically the function of the hippocampus. So when you remember an episode, you're actually reliving it in a way. And here's another interesting clinical tidbit about this binding process that takes place in the hippocampus. Turns out the episodic memory components reach the hippocampus through different related pathways, one through the perirhinal cortex and one through the posterior hippocampus. The aspects of the memory that have to do with identities or the objects are the stationary parts of the episodic memory get to the hippocampus through the perirhinal cortex, whereas the memory itself, the relationship, the context, what happened before what, the temporal aspects of it are dependent on the posterior hippocampus. And as a matter of fact, it turns out that the activation in the posterior hippocampus more reliably distinguishes the ability of a person in determining and differentiating between true events that happened in the past and false events that happened in the past. Whereas the perirhinal cortex sometimes confuses true and false events in the past that are linked to a particular character or a particular object. And this is of clinical importance in the epilepsy world. Nowadays, when we're doing pre-surgical epilepsy workup to prognosticate the cognitive outcomes post-surgically, we look for that posterior hippocampal activation. If it's there, we can somewhat comfortably tell patients that their outcomes post-surgically are favorable. All right, let's learn some lessons from language. And here it is, ladies and gentlemen, the time-honored Wernicke's Geschwind-Lichtheim model of language, with Broca's area being responsible for fluency in the inferior frontal lobe, and Wernicke's area in the superior temporal gyrus, posterior third of it, being responsible for understanding or decoding of language, and the connection between them, the arcuate fasciculus being responsible for repetition. And then you have the angular gyrus and other areas related to reading, and Exner's area related to writing. Here it is depicted as the famous Lichtheim house, depicting the various potential lesions that can happen in this model. You lesion Broca's area, you get a problem with fluency and grammar. You lesion Wernicke's area, you get a problem with understanding. And if you lesion more proximally, you are no longer able to actually pick up words from noise, and you lesion more distally, and you have a motor disorder of speech. And supposedly B is representative of what we call your association core disease, and so on and so forth. This time And here are the different phases of the aphasias that are predicted by this model. Here's a Broca's aphasia, the non-fluent aphasia. Here's the lesion of Wernicke's aphasia, a fluent aphasia with jargon speech. Here's a global aphasia. Here is a conduction aphasia with impaired repetition but intact everything else. And here's a transcortical motor aphasia with a motor aphasia but with intact repetition as opposed to its impairment in Broca's aphasia. And here's an anomic aphasia in someone with a lesion close to the semantic hub, although anomia is by itself not localizable. And this is in no way a discussion of aphasia lecture. But this is to say that the examination based on the classic model persists in having validity. And we have in fact confirmed a lot of its aspects using modern techniques. For example, this fMRI work here reimagined showing that Broca's area is activated when people manage sentences with a higher complexity than lower complexity. Having said that, there's a variability in lesion location leading to the various clinical syndromes that we have, Broca's and Wernicke's, the more classical ones. There's a reimagining to a certain extent of what the model might be. We now think of a dorsal frontoparietal network and a ventral temporoparietal network. The ventral temporal network takes sound, decodes language out of it, and links it to the semantic hub. This is the key of the semantic network, and therefore you deduce meaning. You also, from there, get to the parietal lobe where your phonological buffer is. This is the crime scene where you determine grammar and who did what to where and so on and so forth, the relationships between the characters and the objects in your expression, and you link anteriorly to Broca's area in order to figure out the grammar of what you are going to say. So this unification of with memory gave the model the name of memory unification model of language, because you're linking language with semantic memory. And this may explain the fact that your lesion may be along this pathway or along that pathway, therefore putting us in the modern perception of cognitive functions as being network phenomena, not necessarily specific lesion-based phenomena. All right, a few lessons from perception, specifically visual perception. So visual perception draws our world dot by dot, line by line, shape by shape. And with hierarchical processing, we determine colors and we determine motion and so on and so forth. And we have various areas ranging from elementary visual processing until your VX areas down the line, determining the more specific aspects of the visual percept. Some cells respond to colors better than contrasts of black and white, like those in V4. Some cells respond to movement and speed, like those in V5. And we know this information is recreated from humans using functional MRI. And we know that more ventrally located areas are responsive to shapes and color and details of objects, whereas more ventrally located, more dorsally located areas are responsive to motion. And from that, we now have the two-stream model of visual pathways, with the ventral pathway being the what pathway and the dorsal pathway being the where pathway. And a lesion in the where pathway might impair recognizing objects by movement, so-called achinotopsia. And whereas a lesion down the ventral stream might impair object recognition and lead to agnosias. How do we identify objects along the ventral stream? We first determine shape. Areas like the lateral occipital cortex are especially active when lines of shapes are shown in comparison to static and ambiguous shapes, for example. And areas along the ventral stream are also responsive to, they're able to identify, especially areas on the right side, they're able to identify objects even if you change their position. So you don't have to be exposed to the different angles and shapes of an object. You can still identify the object because it's been recreated for you. And these things happen at higher stages of visual processing. At even higher stages, there is what we call holistic processing, meaning identification of objects not by the specific details and bit-by-bit analysis, but by identifying them as a whole. So there are areas, famous areas for facial recognition. The fusiform face area is well known. The occipital face area is another one. There's also a superior temporal sulcus face area that is responsible to the emotional content of facial recognition. There are other examples of holistic recognition of objects, including the extrastriate body area that responds to shapes of body parts. Even if it looks like a mitten, not like a body part, you'll see a little bit of an activation in that area. Here's the superior temporal sulcus face area that I was showing you and mentioning that it responds to the emotional content during facial recognition. There's also an area responsible for recognizing scenes or pictures of cities or places you've been to, the so-called the parahippocampal place area. So recognizing objects is by no means a simple process. It begins with lines and edges and colors and movement and form and so on and so forth, and then it forms the percept at various levels of complexity It recognizes patterns and then infuses it with meaning by connection to the semantic system. Therefore, object recognition relies heavily on the semantic system as well before linking to language, and you're able to name an object. And in order to test that at the bedside, we have various maneuvers. We have people copy objects, copy pictures. We have the match, and so on and so forth. And the more proximal the problem is in the perceptual process, the more difficulty you'll have with copying objects and matching them by shape. And the more distal it is, the more you'll have difficulties recognizing objects' functions and identifying what it does, and so on and so forth, based on the visual percept that you've already formed. And therefore, we have different kinds of agnosias, apperceptive agnosias, integrative agnosias, and associative agnosias. Once again, this is by no means a lecture where we can delve into the depths of fascination of agnosias that captured the imagination of Sigmund Freud himself. But this is to demonstrate that careful assessment of the neuroanatomy and neurophysiology that we know our head will help us understand our patients better, and will help us know whether this patient has an anomia, whether it's a language problem, the patient can recognize the object but just lost the name, whether it's a memory loss, this is an amnesia, essentially, or whether this is a modality specific. Someone can recognize the object based on its visual properties, but can recognize it based on auditory information, for example. All right, so this was kind of a cognitive marathon, both literally and figuratively, but here are the conclusions. Cognitive and behavioral neurology is strongly grounded in cognitive neurosciences, neuroanatomy, and we are proud of our focus on clinical observation and examination and sorting things out, judging what this patient have. This is much more than the test batteries that we typically use. The cognitive exam is not a MoCA score. And while these tests are important and they help us standardize our testing, they provide cutoffs for dementia, for example, and so on and so forth. They should not replace the clinical approach. Examples are plenty from the field of memory, from the field of language, perception. We mentioned many today, and they span fields of dementia and epilepsy and many other stroke and so on and so forth, you name it. And in order to be able to save all of these patients with quality, we need to work together. We need to create tools that are specific for our patient population, customized for our patient population. We need to work together in order to create research projects that help us understand our patients better and our peculiarities. Neurologists, psychiatrists, geriatricians, psychologists, cognitive neuroscientists, you name it, linguists, working together will help us achieve loftier goals for ourselves and our patients and community, more importantly. And thank you very much from me personally and on behalf of the Saudi chapter of Cognitive and Behavioral Neurology. Thanks again.